First of all, thank you all for coming, and Jackie, thank you for the nice introduction. As Jackie said, my name is Aaron Santos, and I'm going to talk to you a little bit about some of the work that Derek Lyons and I have been doing in the nanotech and biotech space. So I know for the faculty here, a lot of you had some questions about why are you still here? Are you still getting paid? The second question is easy to answer. I'm not still getting paid. But about a year and a half ago, I left Simpson College to go to EMC Insurance because EMC was doing a lot of very innovative things, and it was a really good opportunity for me. But I did not want to leave Simpson completely because there was a lot of very exciting and also innovative things happening on Simpson. Specifically stuff happening through the Emerge at Simpson program, out of which two startups that Derek Lyons and I have put together, DNP123 and Express Seed, they came out of the Emerge at Simpson program. To really understand the kind of genesis of this, of these startups, I have to give a little bit of more personal background just to give some underlying understanding of kind of where my thought process was when these ideas got started. So a while back, when I was an undergrad, when I was your age, I worked at a place called the MIT Media Lab. And the Media Lab is kind of like Willy Wonka's chocolate factory for geeks. And it was started in the 1980s with what at the time was this crazy idea that if you looked at all of these different forms of media, your telephone, your TV, your camera, newspaper, mail, your music, all of those were just different forms of what could be digital media. And at the time, they were telling companies that 20 years from now, you're not going to be looking at a newspaper that you hold that's a physical piece of paper in your hand. You're going to be looking at this on a screen. And they were somewhat ridiculed at the time for that, but they turned out to be very right. So actually, as I was throwing this up there, I realized some of the objects in this screen, some of the members of the audience are going to be too young to know what any of those are. So I'm going to get off this. I'm going to get off this screen pretty quick. So as I was putting this talk together, I actually went back and looked up to try to figure out what the Media Lab was doing now. And I came across a TED Talk by Joey Ito, who is now the head of the Media Lab. He gave a very nice TED Talk that I highly recommend in 2014. And he made a very astute observation in that talk. And that's if you look at the way people used to have to do any sort of innovation. It started with you got an MBA and you paid that MBA to put together a business plan for you. Then you went out and you tried to get some venture funding, so you got your $1 million or $2 million or however much you needed to do the thing you wanted to do, and then you paid some engineers to go and build the thing you wanted. And what's interesting is if you look at what's happening today, at least in the software space, that model is flipped largely because the cost of innovation, because of the internet, is so very cheap that the new model seems to be more, if you look at the Googles and Facebooks of the world, you have an idea, you go out and you build the thing, then you get funding for it, and then kind of later you try to figure out what the business model is going to be for it. And again, this is easy to do in software because the cost of making software is so very cheap that kids in their dorm rooms can go and start the next Facebook. And one of the questions I wanted to ask was, how do we do the same thing in other areas? So how do we enable rapid innovation for biotechnology and nanotechnology? So how can we have it, today, if you watch that TED Talk, Joey Ito talks about how gene sequencing is so cheap right now. There are literally college kids in their dorm rooms that are going and entering synthetic biology contests. So how can we make that even more accessible to people? So that's partly where the idea for DNP123 came from. So at DNP123, we are enabling the rapid design and manufacture of fully functional nanotech products. If you are not sure what nanotechnology means, it's basically just the science of really small things. So nanotechnology is the study and application of extremely small things, about 1,000,000 times smaller than a millimeter. And it's generally used across many different science disciplines. So it has applications in physics, chemistry, biology, material science. Just about any science field you can think of is affected by things on a nanoscale. And when I was first getting into science as an undergrad, nanotechnology was the big thing. If you put nanotechnology on a grant, the joke was that as long as you put the word nano in there somewhere, it was going to get funded. NSF was predicting that there was going to be $1 trillion market for nanotechnology. And what was odd to me is at the time there was this big disconnect between what people were actually doing and then what it was getting billed as. So if you looked at what people could actually do, people were really good at making geometric shapes. So they could got my laser pointer here, you could make cubes, you could make prisms, you could make spheres, you could make carbon nanotubes, which are basically cylinders. You could make different aggregates of these things. You could get them to combine into very symmetric crystals, which to get to just this point was a huge scientific leap and took lots of researchers putting lots of hours in. But what you read in pop science was always nanotechnology was going to give you this. It was going to be nanobots swimming around in your bloodstream, lancing your cancer cells, and then letting you live to be 120. And there's a big jump between nanorobot and sphere, right? Like, if you go into nanotechnology thinking I'm going to get to play with nanorobots, and then you're playing around with spheres, like it's very disappointing. So I tried to figure out like why weren't we making nanobots yet? There was no $1 trillion marketplace, and the reason was even though people could think of many useful devices to make on nanoscale, not just nanobots, but transistors and a whole bunch of other things, there was this mismatch between what you could design and what you could actually manufacture. Simply put, there wasn't a 3D printer. There wasn't something that you could go and just make anything out of. There were no lathes. Making things that are that tiny is just very difficult to do. So we said we're going to combine two different technologies to try to make that better. The first thing we did is we used the programmability of DNA. So you might remember from high school biology, DNA is a double-stranded helix. If you take those two strands apart, what you'll find is that there is a sequence of chemical bases in DNA. There's 4 types of bases. There's adenine, thymine, guanine, and cytosine. And to get that double helix, those bases have to bind together. But they bind in a very particular way. So adenine only bonds to thymine, A only binds to T, and cytosine only binds to guanine, C binds to G. And what that effectively does is it makes DNA a kind of very smart Velcro. So if you have a particular sequence, there is only one other sequence that first sequence will bind to. We call that a complementary sequence. And if you're a biologist, you think of DNA as a code for life. If you're an engineer, you think this is a really cool building material. Because if you're smart and you can figure out how to connect certain sequences to other sequences, all of a sudden you can start to design things. So there was a paper that came out in 2006 by Rothermund that introduced the field of DNA origami, which is basically DNA pieces stapling other DNA pieces together to make a bunch of different cool shapes. So you can make stars, you can make smiley faces, you can make triangles. In one paper I saw they made a map of the United States. Lots of really cool things. The problem is that doesn't really do anything other than look cool. If you actually want to build devices, like an electronic device or an optical device, usually you need different materials. You need metals, insulators, semiconductors, which you also have at nanoscale. People make nanoparticles out of different metals, out of semiconductors, and nanoparticles actually have some functionality to them. So they have different optical properties. So the solution on the left is when the particles are, Derek, you can correct me if I'm wrong, bound together? No? Separate. So aggregated on the right, separated on the left. So when the particles stick together like this, you get a color change. And you can use that to detect different chemical properties. They also have interesting electronic properties. So what we wanted to know is, what if you could take the programmability of DNA and combine it with the functional properties of nanoparticles, since nanoparticles can't be programmed to assemble different shapes. So we had what we thought was a very simple idea. We're just going to take nanocubes, which we know we can make, and different DNA, single strands of DNA, and we're going to try to paint each face of the nanoparticle with a different sequence of DNA. So this purple might be one sequence of DNA. These green molecules on top might be another sequence of DNA. And if you can do that, it allows you to design how the blocks are going to connect together. So if I have two blocks, here and here, and I have a particular sequence on one strand, and then I have that complementary sequence that'll hybridize and form that double-stranded helix on the face of this cube, what will happen is those cubes will basically get glued together. If I do the same thing, I have the same sequence as before, but now I try to combine it with a sequence that is not complementary, all of a sudden you don't get binding together. You just get two cubes that are floating around in solution, and they just kind of stay away from each other. So our main innovation is we figured out how to connect blocks, which doesn't sound like much, but when you can connect blocks, all of a sudden you can start to build up complex structures, right? So taking an analogy from the video game world, if you want to make Mario, that's really just different combinations of 3 blocks. If you prefer Legos, just by building the same, taking the same blocks over and over again and combining them in different ways, you can make very complex structures out of that. In fact, nanoparticles with DNA patched on them would be even better, because if you could figure out how to get the DNA on each face, you wouldn't even need to have to connect them, right? Legos, you have to sit there, and every block you have to add on to the structure. Nanoparticles self-assemble. What that means is if you just figure out which patch has to go where, so which sequence of DNA has to go on which face, and then you just take all the cubes you need, throw it into a jar, shake it up, and come back in a few minutes, the random motion of the particles in solution will make it so that they automatically assemble the structure that you want. So you don't actually have to have any complicated robotics. You don't have to sit there with some tweezers and move stuff around. It automatically forms this. It completely takes away the manufacturing process. So we spent quite a long time trying to figure out how do we actually get the cubes, how do we actually get the DNA on one face. That's not an easy thing to do until we realized we could do something really stupid, which was that if you take a solution of the cube, so generally they're dissolved in some sort of liquid, and then you just take the liquid away, you evaporate it so that you dry the cubes on a surface. Then what you can do is you can take a stamp, paint the DNA you want on the stamp, and then just apply the stamp to the surface. What will happen is only the top portion of the cube is in contact with the stamp, so only the top portion of the cube is going to get that DNA on it. So it gives you a way to coat a single face with DNA, whereas if you tried to do it in solution, all 6 faces would get coated at once. To get the next stamp on, what you do is basically you redissolve these cubes so that they're floating in solution. You dry them again. You put another stamp on. You have to be a little bit careful because there is a chance that what I've drawn here is the red stamp. There is a chance that it's going to land face up again. And you don't want to stamp something you've already stamped. So as we were figuring out how to do this, a little side story here that I find amusing. How many of you guys have either taken or are taking Intro to Prob and Stats at Simpson now? Okay, a few hands. So when you take Intro to Probability and Statistics, they usually give you these stupid problems like you roll a dice 6 times. What are the chances that the dice never lands with the same face up? And I used to think this is such a stupid problem. I'm never going to use this, and this is exactly the same problem. Fortunately, there's a way to get probability and statistics to work a little bit in your favor, so that we know that if we stamp DNA sequence A is going to bind to sequence A prime, its complement. So if you make your surface out of A prime, that A is going to stick face down. So then you know the next stamp you put is going to be on the opposite face. So if you're smart about this, you can do this in a way that gives you actually pretty good yield, even though some of the times you might end up stamping different faces. So you have to do a little bit of purification and a little bit of repetitive stamping. But at the end, you get 6 different DNA sequences on each face of a cube. And as we've discussed, once you can do this, you can make all sorts of unique and cool structures. So when we figured out you could do this, we're like, that's it. We are awesome. Like, we have solved nanotechnology. Like, come give us our Nobel Prize. And we started to show people and thinking that like they're going to think we're really cool. And they're like, all right, well, what are you going to make with it? Right? You said you could make anything. Like, what do you want to make with it? And we were thinking like, well, that's not really what it's for, right? Like, we don't want to solve your problem for you. We're trying to give you a tool that you can go and solve your own problems. But it was clear that that wasn't enough. So I was reminded of the Steve Jobs quote, where he said a lot of times people don't know what they want until you show it to them. So you can't just say, here's some Legos, and give somebody a blank slate and expect that they're going to do cool things with it. It does help to actually show them some examples of cool things you can build. So we decided to go to different industries in the area. And since we're in Iowa, we started with agriculture. And we just went to different industries and we said, hey, what are your pain points? What are things that you think we can solve for you? Or just what are your pain points? We'll try to solve them for you. So we talked to a few different seed companies, and they came to us with a problem, and they said, all right, we've got this issue. We have seeds that we want to sell to farmers. We've got certain genetics that we want in these seeds, and the price is going to depend on what those genetics are. So we really need a way to measure the genetics of different seeds. And just to give you a little bit of background on the market, they said, all right, well, for the market we're trying to get into, there's about 15,000 grain elevators, and usually you see trucks lined up outside of them, so you need to be able to do this fast. You need to be able to do one genetic test about every 10 minutes, and you can charge up to $100 per test. This had never really been done before. So the standard way of doing genetic testing is you use some type of PCR, which is usually in the order of like an hour or so, and it takes big, expensive equipment that's hard to interact with. So this was kind of a tall challenge. But then we kind of did the math on what it would be like to make $100 15,000 times every 10 minutes. And they were like, all right, we're gonna play around with this for a little bit. And not only that, but that's one market. That's one market in for a particular industry in agriculture. It's only for one type of seed. It doesn't get into the other areas of agriculture. It doesn't get into forestry. So things like you could determine in advance if a tree had a certain disease like oak blight, which is causes huge economic problems for cities that have to take those down or replace them. It doesn't get into livestock. So if you're a farmer or a rancher rather, and you are trying to figure out which cattle are you going to breed and which are you going to sell, if you can get that test done within minutes, that is another couple of weeks that you don't have to feed those animals. It saves you money and it makes the whole process a lot more efficient, improves food efficiency and food security. On the human side of things, how many of you guys have heard of superbugs before? Okay, big problem right now with bacteria that if you give it antibiotics too often, they become resistant to it, and all of a sudden, those antibiotics don't work anymore. So there's a fear that we're going to go back to a dark age where penicillin, amoxicillin don't work, and infant mortality rates rise. Well, what if you could, every time you go to the pharmacist at CVS, where you have the cold or flu, you could do a quick 10-minute test to figure out what strain of the flu you had, and then base the specific genetics, base the specific treatment on the genetics that you have, so that you know ahead of time if there's antibiotic resistance, so that you don't want to apply those antibiotics to those flu strains. Huge problems we could solve here. So that we decided to do another spin out of DNP123, which was Express Seed, and that was for fast, affordable, and portable genetic detection. Again, using the DNP technologies. So we came up with a simple scheme that we thought we could detect gene sequences with very quickly. Basically, all we did is we took a bunch of the cubes, stuck them in a row, so we have like a baseball bat of cubes, just a long line of them. And around each of those cubes, we put DNA that was complementary to different parts of the genomic sequence we were interested in. So basically what would happen is if the gene we wanted to detect was in there, it would stick, different parts of that gene would stick to these rods, and it would glue them together in parallel. And if the gene wasn't in there, basically the rods would just kind of float around. It'd be like you took some pencils and threw them on the floor. They would just kind of go everywhere randomly. So that was what we were trying to do. And then we went to lab, we messed around a little bit, and we were able to get this. So when no gene was present, you can see there's not really any parallel lines here. They're all a little bit curved. When the genes were present, you get these very regular arrays with a particular spacing. Again, caused because the gene is there just stapling the rods into this nice ladder structure. Now, again, nobody had ever done this kind of genetic detection before. Normally, this would take a research team at an academic lab or an industry lab. They'd get some budget for it. They'd spend many months, if not years, trying to put something together. How long do you think this took us? Under 2 months. So we had a presentation we had to give in August. We started in June. In 2 months, Derek Lyons came out of a lab with this, a device that showed proof of concept that we could actually measure this stuff. And I remember it because Derek came out and he was all excited, and he'd been in lab for like months. He had this big bushy beard. His hair was all gray. I mean, I'm exaggerating for comic effect, but it wasn't that far from the truth. We would never have been able to do this if we were working on some sort of complicated system where we had to do fundamental science every time. And a good chunk of those 2 months were actually figuring out how to get the cubes to stick together. But when all you have to do is connect blocks to build new and interesting structures that solve problems, all of a sudden solving those problems becomes a lot easier and a lot faster. Not only that, but Derek was also smart enough to put some cellular technology so that the device can actually communicate the results back. So you could imagine that any farmer in the field right now can go take a device, measure the genetics of it. All of a sudden, those results will automatically get uploaded to a cloud somewhere where you can build predictive models. You can start to say, hey, what sort of action should you take to maximize your yield? If you had access to lots of farmers who are contributing to a database like this, you could help them out a lot by trying to predict, based on weather and other things, what the best course of action they could take at different points in time would be. Are any of you guys working on the Palmer Amaranth project? Okay, one of you guys over there, a couple in the back. If you could, it's one thing. So for those who don't know, the Palmer project is tracking the Palmer Amaranth weed, and they're building models that predict its spread. What would happen if you could actually measure where it is and get real-time data coming in of where that's going? Right, you could build much better models much more quickly. So our current state right now, we've got proof of concept. We are improvising and optimizing based on what the industry groups are telling us they want. Also trying to improve some features of the device, like the getting better alignment of the card and that kind of thing. It's handheld. It's simple to use. You basically just press run, and the device will automatically cycle through and tell you if a gene is there or not. It works in roughly under 10 minutes. We expect that we can sell these for as little as $10. It's cloud connected so that you can get that automated reporting and analytics. And we're currently discussing with a few Fortune 500 companies about what we can do to tweak the device to make it best work for their needs. So that's Express Seed. The last thing I want to talk about is what's kind of next for DNP 123. So really the question we're interested in from here is how do we get the technology into the hands of builders and entrepreneurs, people who are actually going to make cool things with the devices. So when thinking about the future, I went back to some of my old PowerPoint slides, and I pulled this from 2014. And I'm amazed because when I first presented this, I exaggerated. I said it was a slightly optimistic timeline, thinking that was an incredibly optimistic timeline. But we actually weren't far off. In 2014, I said in 2 years, we were going to have proof of concept. In 4 years, we were going to be building complex structures. And in 5 years, we were going to be patenting functional devices. We sent out the provisional patent for the genetic detection device in 2017. So we're actually a little bit ahead of time for where we expected to be. So then the question is, what's next? How do we get the device to people who can actually use it and build cool things? In planning to get this distributed, we did a lot of reading and background about disruptive technologies, and I really liked this quote by Clayton Christensen at the Harvard Business School, defining what, in his mind, defines a disruptive innovation. And he said, a disruptive innovation is not a breakthrough innovation, in that it has a very specific definition. And that is, it transforms a product that was so expensive and complicated that only a few people with a lot of money and a lot of skill had access to it. Today, if you want to make any sort of nanotech device, you have to own like a clean room or a big chem lab, and you have e-beam lithography or some other type of lithography that's going to make silicon chips. We don't want people to have to do that. We want to make it so that just like in the 80s and 90s, people were doing startups in their dorm room making new software, we want to have people doing that with nanotech and biotech. So I think there's 3 things that we can do to make it so that more people can be doing this for cheaper. Step 1 is enable play. So we kind of jokingly put this nano assembly kit together, and then I started thinking more seriously about it. So we do have, these are kind of little template flyers that we're messing around with. But the basic idea is we want to give people a little Lego set that they can use and play around with. So what we'll do is we'll give them a few different tubes of different types of nanoparticles with different DNA on them. And then we'll give them instructions for if you mix the cubes in different ways. Here are some sample things that you can make, but really it's up to you. What do you want to make out of it? See how you can connect the cubes. So part of this is because we want to show researchers and innovators, both in academia and industry, that this stuff is really easy to use. Hey, look, even a 10-year-old can do it and make cool and interesting things. But also, we haven't really lost our academic roots. And so there was a night in the middle when Derek was growing his beard out that he called me really late at night and said, this was the most fun day of science I've ever had in my life. And he was really excited. And it was the day he realized that you could actually watch these things assemble and build and take shape under a microscope. You could see them moving around, kind of jiggling, undergoing Brownian motion. And it was that kind of aha moment, that real level of excitement. I want kids to have that. I want researchers to have that so that they know this technology is real, you can build with it, and you can do cool things. So that's step 1. We want to enable people so that they can play with things and feel comfortable experimenting and innovating. Step 2 is it's one thing to mix chemicals around. That gets really boring after a while. So we want to make manufacturing even easier. So we want to improve the design and manufacturing efficiency, enable data collection and sharing, and from that, build a knowledge base with a library of structures and their properties. That's a lot of words. What does that actually mean? So it's one thing to mix cubes together. It's another thing, to make an analogy with 3D printing, you could go and glue a bunch of sticks together, or you could just design that in CAD and push a button that says assemble, which is what you do with a 3D printer. We want to make a CAD version for nanotech. So the idea would be you'd have different cubes and different shapes that you could just pull, drag and drop into a 3D CAD interface. So just like you might design something in Google SketchUp, you can design nanotech things in a DNP CAD software. Of course, you wouldn't just want to have the software. You'd actually want to have a printer to go along with that, so that as soon as you're done designing something, you can just hit assemble, and then it automatically assembles the nanostructure that you've designed, assuming you have the right cubes and ink and such. But going back to what Joey Ito said about students building the next biotech startups in their dorm room with desktop gene sequencers, we want Simpson students and other students to be able to go in their dorm room, design new products, and then showcase them to other people. So, if somebody says, "Hey, I'm a business person. I'm interested in what you're making," you can just take their business card, spit some stuff out, and show them what you've made. Say, "Hey, go look at this under a microscope." The other thing we want to do to make the manufacturing process even easier is help makers take devices to market. So the Merge at Simpson program was great with getting us the resources we need to actually set up a company. So things we don't know, like any sort of legal or other ramifications for running a business. They took care of all of that on their end. To the extent that we can help other startups and get them up and running so they can start using our technology, we want to do that. And this is kind of my toy idea, but I really want to make nanotech Etsy. So the idea is that you design your structure, and if you have a community of people who are building different things, and person X is like, "Hey, I made this widget." Well, all you have to do is just drag and drop that widget. Maybe it's an electric motor or something on a tiny scale. You drag it into your interface. You're like, "Hey, I want to use that motor in this thing I'm building." You drag and drop, and then Doodoo made the motor gets a couple of bucks for selling it. So basically, nanotech Etsy. And the last thing we want to do is really automate the design process itself. So the old school way of doing science is you have people in a lab mixing chemicals together. For the most part, that's not the most efficient way to do things anymore. So a lot of industry labs, it doesn't look like that at all. A lot of these experiments that you used to have to mix by hand, there's robotics that will do that for you. So it becomes a lot less like this and a lot more like this, typing at your computer, working from a coffee shop, designing your next cancer drug or whatever it happens to be. So if it's going to be this anyway, why don't we use machine learning and data science to do an even better job than what people could do just trying to think, how do I connect blocks together? And there's, using machine learning, there's a lot of examples of how AI can solve problems better than humans can, at least very specific problems. So Ken Jennings getting beaten by, Deep Blue in Jeopardy. I have another example I'll show you in a second. But the basic idea is, rather than design the structure ourselves, we really just want to specify what do we want it to do. And if we can define the task, we can use machine learning to define the optimal solution that nanoparticles will use to solve that task. So what do we need to know? You need to know the state, the configuration of the system. So what cubes are stamped where? What is that going to assemble? We need to know what actions the assembler is going to be able to take. So for us, that would be make cubes, dry the cubes on a surface, stamp the cubes with DNA, redissolve the cubes, mix cubes. There's only really like 10 things we do in a synthesis. We just do them over and over again. And then you need, this is the tricky part, is you need some way of giving a reward or some sort of point system for how close you are. So let's say you want to make a drug delivery device. Well, you need to say certain devices you build are closer to what you want than others. So you need a point system for assigning how good the structure you've made is. And then the output of the algorithm is just the policies you get. So that's what action do you take at any given state. So if I have a structure that looks like some shape, do I put more cubes on it? Do I stamp again? How do I work with that? So again, that's very abstract, and I wanted to give you guys at least a concrete idea. And mostly, I just love this video, so I wanted an excuse to show it. This is the CEO of DeepMind Technologies, and we're going to hope the audio works on this. It's just a minute long. So what they did is, using the same machine learning algorithm, they basically just trained it to play video games. But you'll see they're a lot better at playing video games than most people are.
So I'm just going to run this video now. This is a 1-minute video. This is Space Invaders, the most iconic game probably on Atari. And this first minute, this is before the first time the AI has ever seen this data stream. So don't forget, it doesn't know what it's playing, it doesn't know what it's controlling. And you can see it's actually losing its 3 lives. It's controlling the rocket here at the bottom of the screen. And it's losing its 3 lives immediately because it doesn't know what it's doing. But after you leave it playing overnight on a single GPU machine, you come back the next day, and now it's superhuman at the game. It's learned for itself through experience how to play. So you can see now every single shot it fires hits something. It can't be killed anymore. It's worked out that the pink mothership that comes across the top of the screen in a second is worth the most number of points. And it does these amazingly accurate shots to do that. And you can, if those of you remember Space Invaders, as there's less of them on the screen, they go faster. And just watch the last shot that it does, the rocket does. There's this predictive shot to hit the last Space Invader. So you can see how perfectly it sort of modeled the game world and that data stream. So accurately, it can predict ahead of time what is going to happen.
So video games is a little bit of a jump from nanotechnology, but the algorithm's almost exactly the same. Right? You've got a handful of buttons that you can press. In this case, it's a little bit more buttons because it's a Nintendo controller rather than Atari. But all you're doing is trying to predict the sequence of things that you do. All right, so do I press the up button, the B button, the A button? There's an algorithm, and there's libraries of algorithms that you just download and you run, and they will solve that problem for you using deep learning algorithms. Similarly for us, if we just had a way to, a similar way to a point system like you have in the Atari game of measuring how close you are to different structures, well, again, we only have about 10 possible things we can do. We can make cubes, we can stamp cubes, we can dry them, we can suspend them. It's the exact same problem in a different way. So just using the same algorithm, we should be able to design devices. In fact, we don't even have to design them. We can let the AI do that for us. So for my final future thought, I'm tacking one more thing onto my schedule here. I want to have AI designed devices by 2022. So we'll see if I'm still accurate by then. Final thoughts. DMP-123, we're enabling the rapid design and manufacture of nanoscale products. We're already designing and making products through Express Seed. We're certainly looking for students and innovators who want to make products, want to design products, and want to mess around with this. And fun stuff is coming. CAD, nanoprinters, AI. So with that, I am going to stop here, and I will take questions.